Haystack Analytics, introducing Haystack Labs. And we have series of webinars organized for general NGS sequencing services awareness. And so today's seminar series, we will have a broad idea about the seminar series is sequencing landscape, a full spectrum of sequencing technologies from short read to long read sequencing. Let me introduce Haystack Analytics. Haystack Analytics is a health tech company based at IIT Bombay. We are a bunch of IIT graduates who were passionate enough to bring the NGS solutions in solving real life problems. We have tuberculosis genomic diagnostic solution, which is recognized as one of the top from BIRAC. We also have a flagship product called Infection, which is pan bacteria, pan fungus, and clinically important ARGs detection from the samples. Our vision is to transform healthcare through genomics. To highlight our focus is to fulfill the gap of healthcare by creating scalable solutions to benefit patients in the overall healthcare ecosystem. We have our partners as Intel and GE Healthcare. We are supported by one of the major funding agencies in India and the universities in India. Our partners are major hospitals in India and the labs. Let me briefly take you through the journey of Haystack. Haystack as a company was incorporated in 2019, and as we got incorporated, we also were awarded Maharashtra Startup Award with the Maharashtra Startup Award, followed by which we have created a solution for TB using genomics, which is now validated, which got validated with the NTP. In 2021, we have launched TB Genomics Pan India, and we are actively serving the TB population with a 18 drug DST report and mixed infection or NTM. We have also done in 2021, where all the world was focusing on COVID. We also did COVID genomics for the government. In 2022, we won the BIRAC big grant for TB. And then we also initiated the development of our universal infectious disease test, which is Infection. In 2022 itself, since we are working actively in developing state-of-the-art and validated reproducible softwares for genomics data analysis, our Haystack Genomics platform got integrated and used by Intel and GE Edison Award. In 2023, we have got again the Pan India launch. Let me introduce you to the speakers for today. We have Harold, who is field application scientist from MGI. We have Dr. Ravi Kumar Chilukoti, who is head India and South Asia technical support from MGI. Myself, Dr. Amrutraj, I will be moderating the talk. I will introduce Dr. Ravi Kumar Chilukoti. He is qualified as a PhD and postdoctoral studies in molecular genomics from University Medicine, Greifswald, Germany. He has around 22 plus years of cross-functional expertise in techno-commercial. He has been instrumental in scientific support, operations, business development, and also involved in genomics-based research projects and diagnostic applications. Our next speaker is Mr. Harold. He is field application scientist from MGI, and his expertise in technical support, training, and application development for MGI NGS technologies. I would like to hand over the stage to Dr. Ravi Chilukoti for his presentation. Thank you.
Thank you, Dr. Amrutraj, to invite us for this webinar. So, hello, good afternoon to everyone. So, this is Ravi representing MGI in this webinar. So, I'm responsible for the technical support and also product management from India and also South Asian countries. So today in this webinar, we would like to introduce MGI C-call technologies developed by MGI, especially the C-call technologies. C-call means it's a portfolio of the technologies developed by MGI for the manufacturing of the short-read sequencers and also long-read sequencers, which are supporting almost all the applications. So this is the content of my presentation. So before going to the presentation, so here you can see there are various NGS technologies emerging from 2005 to till date. So starting from SBS from Illumina, long-read sequencers from Oxford Nanopore and also PacBio and MJ platforms. So especially MJ has started the manufacturing of the short-read sequencers using the DNB-seq technology. DNB-seq stands for DNA nanoball sequencing. So before MJ has started the manufacturing of the short-read sequencers, actually the cost of the sequencing was really very expensive across the world, especially in India also. So once MJ has entered into this manufacturing of the short-read sequencer, the cost of the sequencing is really driving down. At the moment, we are offering $1 for 1 GB using an instrument called T20. This is the instrument T20. So you can compare. So NGS is already, I think most of you are already aware that NGS is being used as a major tool in the era of the precision oncology and precision medicine. So whereas the long-read sequences also now they are started being used and also the optimized ready to end-to-end workflows are being developed for the long-read sequences also to be used for the clinical samples. So coming to the short and long read sequence modalities and what are the major advantages and what are the benefits and also what are the major differences between these two modalities. First of all, the short read sequencer. Here you can see the by name itself short read sequencing means the sequencer cannot sequence the long DNA molecules. Obviously, once the DNA is extracted, DNA or RNA, so they have to be fragmented by using the enzymatic fragmentation or maybe mechanical fragmentation. Okay, so once the fragmentation is completed, then we ligate the adapter and barcode. Then the library will be converted into the DNA nanoball, especially for MGI. So then they can be sequenced on DNB sequencing platforms. Okay, so once the sequencing is completed, so the short-read sequencing data will be highly useful for the mapping, alignment of the data to the reference genome, whereas the short-read sequencing will not be highly helpful for the de novo genome assembly. Okay, so once the reads are mapped to the reference genome, then if you see that there will be various gaps will be there. So these gaps will be highly identified, especially in the regions where the repetitive sequences are there, because short-read sequencing cannot capture the repetitive sequences, especially. So that's the major difference, major disadvantage of the short-read sequencing. Whereas in the long-read sequencing, here you don't need to fragment the data. Once the DNA or RNA is extracted, then we just need to convert into the library by just adding a adapter sequence, and then especially for the nanopore long-read sequencing technology, so we need to just add a adapter because the motor protein will be binding to that one. So once the sequencing is completed, this long-read sequencing data can be highly useful for the de novo genome assembly, because especially if you need to identify the complex structural variants, this long-read sequencing through the de novo genome assembly will be highly helpful. And here also there will be some gaps will be there, but it will be very low compared to the short-read sequencing. Okay, so the long-read sequencing data can be used even for the mapping to the reference genome, and also it can be used for the de novo genome assembly. So especially those species which doesn't have the reference genome, this long-read sequencing data can be used for the de novo genome assembly. So that means we can construct the genome assembly through the overlapping reads by using the long-read sequencing data. It doesn't require the reference genome. Okay, so that's the major advantage of the long-read sequencing. That's why it is being highly used for the research projects, especially for those species which doesn't have the reference genome. Okay, so here in this slide you can see the difference and benefits of the short and long read sequencing modalities, and also what are the major limitations between these two. So as I already mentioned, the short read sequencing data, it is being used since long. So that's why the long history of the usage is there. So because of that, the standardized workflows starting from the sample to the report workflows are already available for almost all the applications. So that's why these are highly helpful for the accuracy data. So you can get the very high, accurate data. So and also at a very low cost of the sequencing, you can sequence with high depth of the sequencing. So this will be highly useful for very accurate data compared to the long read sequencing, especially for the nanopore technology. Okay, so the major limitation of the short read sequencing means already, as I mentioned, it cannot sequence the long DNA molecules, and apart from that, it cannot capture the very complex structural variants, especially the long copy number variants, and also some fusions and also it is very difficult for the short read sequencers to capture the pseudogenes and also long STRs. Okay, short tandem repeats, and also HLA regions also sometimes it will be very difficult to capture them. And so coming to the benefits of the long read sequencers, so the long read sequencers, instead of the short read sequencing, whatever it cannot capture, it can easily capture that one. So for example, the complex structural variants and also the fusions, pseudogenes, STRs, I mean the STRs which are having very long STRs also. Okay, so the STRs means short tandem repeats can be captured by the short read sequencing if the STR is small, whereas if the STR is very long, the short read cannot capture it. So whereas the long read sequencers, they can easily capture the short and also long STRs also. And it doesn't require to prepare the library. So it's very simple to prepare, and immediately the sequencing can be completed, and it can easily capture the very complex structural variants also, and also fusions. Okay, so then especially the real-time identification of the methylation and also metagenomics and also direct RNA sequencing can be done by the long-read sequencers. Whereas the major limitation of the long-read sequencers is, especially it is having very short history of the usage. That's why, especially for the nanopore technology platforms, the standardized workflows for the clinical samples are very, very few, very, very low. So that's why majority of the diagnostic companies are for the clinical samples, the long read sequencers are really not being used. So whereas the quality of the data is also not, I mean, it's good, but it is very difficult to achieve Q30 or Q4, especially for the nanopore technology platforms. Whereas the PacBio platforms, they can easily achieve the high quality of the data, even Q40 also, but the cost of the sequencing is very high in the platform, in the PacBios. Okay, that's the reason. So if you want to have high quality data using the long read sequencers, then you need to use the PacBio platform, but the cost of the sequencing is very high. So these are the major limitations. So that's the reason why the short read sequencers are being used as a workhorse for the standard analysis for the clinical and also research projects. So whereas there is a new modality, combined short and long read sequencing modality. So this is so most of the publications, they are mentioning that the combining of the short and long read sequencing data will be the best workflow for the comprehensive genome assembly analysis, especially for the species which doesn't have the reference genome. Okay, so we can easily get the very accurate discovery of the genetic variation. So whereas wherever the short read sequencing is failing to capture the sequences, I mean regions, then the adaptation of the some of the long read data will be helpful to capture them. So that's why this combination of this short and long read sequencing data will be highly useful as a workflow to construct the complex genome assembly of the species which doesn't have the reference genome also. So here you can see some of the publications which are already published using adaptation of the some long read sequencing data to the short read sequencing data. For example, here you can see in the Parkinson's disease, the long read sequencing resolves the complex structure variants by adding to the some of the short read sequencing data. And one more case also. So here you can see even nanopore sequencing platform data also can be adapted to the short read sequencing to resolve the complex structural variants. And it can be used even for the human cancer genomes also. So especially for the detection of the complex structural variants in the cancer samples. So this way, this combination of the short and long read sequencing data is highly helpful even for the metagenomics and also cancer samples. So this way, the combination of the short and long read sequencing modality will be the best workflow for the construction of the complex genome assembly by using the de novo genome assembly. So coming to the CCAL, so that's the reason why MGI has started manufacturing of the short-read sequencing data by using the DNB technology. So my colleague Herald will explain the workflow of the DNB sequencing technology, and also how you need to construct the libraries and how the sequencing will be done. Everything will be explained by my colleague. Okay, so that's why I'm not going into the details of this workflows. Okay, so once we have started manufacturing of the short-read sequencing platforms using this DNB-seq technology since 2017 onwards. So what does it mean by DNB-seq? DNB-seq stands for DNA nanoball sequencing. So later, just 1 year back, we have started manufacturing of the long-read sequencing platforms using a technology called cyclone-seq. This cyclone-seq technology has been developed on the basis of the narrow principle only. Okay, so I will explain in G series, the G99, it is having two slots. So that means you can run one flow cell or two flow cells at a time. So this G99 is the ultra fast platform because even the patent 150 read length flow cell also can be sequenced within 12 hours. So in 12 hours, you can complete sequencing of two flow cells. So each flow cell of the P150, it can generate 24 GB data output minimum. Okay. So in 24 hours, the G99 can sequence 4 flow cells of the P150. So this way it can generate minimum 96 GB data output within 24 hours. Okay. So that's why it's a ultra fast platform. And in the G50, it is the one of the old platform which was released into the market in 2018, I think. So the G50, the major data output will be 150. So here we are providing two kinds of flow cells, small flow cell or large flow cell, depending on your sample volume or depending on your data requirement per sample. Okay, you can decide to use either small flow cell, which is generating the data output of up to 30 GB, or if you have multiple samples and also if you require higher data output, then you can use the large flow cell large, which can generate up to 100 GB with the P100 flow cell or 150 GB maximum with P150. Okay, so the G400, this instrument is one of the best platform performing very well since long, and also this G400 is being used by multiple service providers across the world. So the G400 also similar to the G99, it is having two slots. You can run either one flow cell or two flow cells. Here also we are providing two kinds of flow cells, small flow cell, large flow cell, depending on the either sample volume or data output. You can decide either to use small or large. Okay, depending on the data output. So if you run two flow cells at a time, the G400 can generate up to 1.4 TB data output. That means 1440 GB data output in full run. Okay. So this way, this will be highly helpful for the service providers or those clients who would like to sequence the whole genome of the human samples or plant samples also. Okay. So this will be very cost affordable, especially for the whole genome also. And coming to the T series, there is one more platform in the G series which was released just 1 month ago. That is called T1. I will show you in the coming slides. Okay. So in the T series, we have designed two platforms, T7 and T20. So these two are the floor standing instruments. So the T7, it is already purchased by the organizer Haystack. They are already successfully using it for multiple applications. So the multiple corporate hospitals and also some of the diagnostic companies, they are outsourcing the samples to Haystack to get the sequencing of whole exome, whole genome, and also metagenomic samples, multiple samples. So this T7, this is really one of the best platform that can be helpful for the service providers who are having the sample volume more than 1000 per month. Okay. So this T7, it is having four flow cells. It can run either one flow cell to up to 4 flow cells. Each flow cell, it will give you the flexibility to sequence to target the data output of 1.2 TB to up to 1.7 TB. So it is also ultra fast platform. In 24 hours, it can generate 7 TB data output if you run 4 flow cells. Okay, so in the T20, this is one of this has been designed by MGA for the countries who would like to run whole genome sequencing of the population. So those countries who are planning a project to whole genome sequencing of the population, this will be highly helpful because in full run, you can the T20 can sequence 6 flow cells at a time. You can run 576 whole genome samples in full run in one run. Okay, so this will be only helpful for the population genomics, for the whole genome sequencing of huge number of the samples. So whereas if you need to run small panels like whole exome or those things, it will be not helpful. Okay, so in the long read sequences using the cyclosec technology, we have designed two platforms, WT02. So here by name itself, 02 means it can run two flow cells. Okay, so each flow cell, it will generate, if you run the flow cell continuously for 72 hours, it will generate 50 GB data output minimum per flow cell. So if you run two flow cells, it will generate 100 GB data output. Okay, so whereas in the WY01, which was released just 1.5 months ago into the market, so it can run only one flow cell, but it has been designed for those clients who would like to run whole genome sequencing of either human, plant, or animal, because it will generate in one flow cell, it will generate 400 GB data output. So this is the information about the genetic sequences designed by DNVSec and also cyclosec technologies under CCAL portfolio. So here in this slide, you can see the comparison of the specifications in the shorted sequences starting from E25 to T20. So here you can see the applications, except the E25, the remaining all platforms, they can run all the applications, including the whole exome also. But the G99 and G50 for the whole genome sequencing of the human sample, it will be expensive. That's why, except the whole genome sequencing, they can run all the platforms. Whereas the G400, T1, T7, these are all will be highly cost affordable for the whole genome sequencing of the human sample also. Okay. So, and apart from that, these three platforms, G400, T1, and T20, they have been being used for the novel applications like single cell RNA sequencing and also spatial transcriptomics, which require higher data per sample. Okay, so that's why these three platforms will be highly cost affordable for that kind of novel applications like single cell RNA sequencing or spatial transcriptomics and spatial proteomics also. And what kind of read length we are providing in the shorted sequencer? We are providing the read length starting from SC50 to SC400, okay, for the single end. Whereas if you look for the paired end, we are providing the read length starting from PE50 to PE300. Please note, in the single end, 50 base pairs to up to 400 base pairs. In the paired end, 50 base pairs to up to 600 base pairs through PE300. Okay, so this way we are providing multiple read lengths flow cells. So wherever you can run any kind of application, for example, 16S rRNA, V3 and V4 region, and also 4 and 6 samples and STRs who are having if you need to identify the STRs up to which are having a length of up to 400 base pairs, you can run it on SE400. Okay, so this way, these applications, these platforms are highly helpful for all the applications, including the long length of the samples. And apart from that, what is the runtime? So the runtime will be very low, except this G400. Okay, so that's why the G4 we have released the T1, which will be very ultra fast platform. I will show you that one. So here the runtime means turnaround time of the sequencing means, except the T7 and T20, all the platforms starting from E25 to T1, the runtime of the sequencing includes the demultiplexing also. That means once the sequencing is completed, because if you are running multiple samples, each sample will be added with a barcode or index. Okay, so once the sequencing is completed, the instrument software itself will be identifying the reads that are coming from sample 1 to sample 2 by using the sequencing of sequence of the index. So that is called demultiplexing. The demultiplexing also will be done by the instrument itself, starting from E25 to T1. Okay, so that's why the runtime is including the demultiplexing, whereas the competitors, they can't do the demultiplexing, especially now they started including the demultiplexing also into the instrument. But since beginning, MJ has providing the demultiplexing also inside the instrument itself, onboard in demultiplexing. Okay, so even the T1, T7, once the sequencing is completed within 22 hours, within 2 to 3 hours, the demultiplexing can be done. So total 25 hours, starting from sequencing to demultiplexing. Okay, so that's why you don't need to worry about the getting the raw data. I mean, FastQ files, even T7 also. So apart from the sequences, once the sequencing is completed, you need to analyze the data. So most of the diagnostic service providers who are purchasing instruments, they are worrying about what kind of applications or pipelines are required, And because most of the times you need to upload the data to the cloud-based analysis. So that's the reason why we have designed the offline bioinformatics servers. For example, the Megabolt, Gbolt, and also Gtron, these kind of platforms. So these servers are offline, and you don't need to upload the data. So these servers are having the hardware called FPGA, Field Programmable Gate Array, which can accelerate the data analysis of whole genome and whole exome. For example, the Megabolt and Gtron, they can complete the whole genome sequencing secondary analysis within 2 hours. Okay, whereas if you need to sequence the analyze the whole exome data, for example 10 TB data, 10 GB data, it can complete within 20 minutes. So this way, the FPGA, the field programmable gate array, which will be helpful for the Megabolt and also Gtron to accelerate the data analysis of whole genome, which can be completed within less than 2 hours, and whole exome less than 20 minutes. And apart from that, nowadays, most of the service providers in India and also hospitals and also some of the diagnostic companies who are having collecting multiple samples with different applications. And obviously, if they need to prepare the library preparation manually, it's very difficult for multiple samples, right? So that's the reason why we have designed the automation system also since beginning. So these automation systems which we are designing, starting from the sample transfer to extraction of the DNA and RNA are both, then automation of the library preparation using SP100 or SP960 and also Smart 8. So these liquid handling systems, they are having the ready-to-use scripts to automate the library preparation using MJ kits and also third-party kits also. So that means these automation systems, they will be helpful not only for MGA platforms. If you need to prepare the libraries using the Illumina protocols or Twist protocol or Twist kits or maybe Roche or IDT, Agilent, everything. So we have the ready-to-use scripts for them also. That means those clients who are having the Illumina platforms or Thermo platform, any kind of platform, they can use these automation systems. Okay, you don't need to worry about purchase the instrument of sequencer also. So without having the sequencer also, if you need to prepare the libraries yourself, and if you want to automate the library preparation, you can use this liquid handling system, which can be helpful for preparing the automation of the libraries by using MJ kits and also third-party kits, any kind. And now, since last 3 to 4 years, most of the researchers, they are using novel applications like single cell RNA transcript and also spatial transcriptomics. So we enter into this one also. We designed a droplet generator, automated droplet generator for the single cell RNA preparation, because once the cell expression has been prepared, the cells should be encapsulated within the droplet. So we have designed this DNB6 CTM4, which will automate the droplet generation by encapsulating the cell and also cell beads. I will show you that one in coming slides. And apart from that, regarding the spatial transcriptomics, you don't need to purchase an instrument. Large tissue sections like human brain or human heart, this kind of chips are not available from any other company. Only MJ is the only one which is providing the chips field of view starting from 0.5 square centimeter to up to 30 square centimeter. I will also show you that one in coming slides. So just for an example, this CCAL product portfolio, what are the product solutions that are available for precision oncology and precision medicine, either from MJ and also third-party company. So here left side, you can see the exome panels. We have designed our own panel that is called V5 panel, that is covering around the panel size is 69 MB, which is covering almost 33,000 genes. Okay. And apart from that, nowadays, most of the diagnostic companies in India and also service providers, they are using the third-party companies like Twist Exome 2.0 or Agilent Tissue Select and IDT Roche. So that's why we partnered with them. So they have optimized their panels to be sequenced on DNB6 platforms. So that's why you can use third-party company libraries also for the exome sequencing. Okay. And regarding the oncology panels, there are multiple panels designed by third-party companies and MG also. So these panels are specifically designed either for only one cancer. For example, this BRCA1, BRCA2, which is specifically designed for the ovarian and also breast cancer. And also nowadays, the medium pan cancer panel, that means covering up to 100 genes. And also, for example, we designed a medium pan cancer panel that is covering around 53 oncogenes and tumor suppressor genes. And apart from that, we partnered with third-party companies like Sophia Genetics and also Paragon Genomics, Pillar Biosciences, which they have optimized their panel to be sequenced on DNB6 platforms. So that's why you don't need to use MJ kits only. You can use the third-party company kits also to be prepared, because MJ is providing a specific chip flow cell on the DNVSec platform that is called YAP-A or YAP-C or YAP-D flow cells. These flow cells will be useful once you because those clients who are already having the SOP to prepare the according to Illumina library, they can prepare the library according to Illumina library. They don't need to change the SOP. Once the library is prepared, if they need to sequence on MGI DNVSec platform, we are providing a conversion kit because our final library is the single stranded circular DNA. So whereas the Illumina libraries will be double stranded DNA libraries. So the double stranded DNA library will be converted into single stranded circular DNA by using the conversion kit. Then the Illumina libraries can be sequenced on DNVSec platform. So this kind of conversion kits are being provided only by MJ, not any other company is providing. Okay, so and nowadays for the oncology samples, most of the oncology clinicians, they are suggesting to go for the comprehensive genomic profile panels. Okay, so which can report even the TMB, tumor mutation burden, and also microsatellite instability also. Okay, so for the immunocheckpoint inhibitors. Okay, that's why we partnered with multiple companies. For example, here you can see one of the company, Gene Plus, which is having a very huge comprehensive genomic profile panel that is covering around 1021 oncogenes and tumor suppressor genes. So this is one of the best panel where other companies they are not able to cover these many genes, oncogenes and tumor suppressor genes. Okay. So regarding the single cell and also spatial, the technology is called StereoSec. I will explain one of the slide because in future also my colleagues, Haystack, they're targeting some webinars, so they will explain that one also. So coming to this CCAL, it provides the end-to-end solution for clinical and also research applications, and it makes omics very more comprehensive, and also it will generate the very accurate data, and also it is very cost affordable. So that's why this CCAL portfolio will be helpful for the public to receive the very rapid and also precise diagnosis reports and also treatments. So I'm just showing you one example here. What is the end-to-end solution for the cancer diagnosis using our platform, for example. So starting from the DNA extraction to library preparation, either by using the multiplex PCR-based panel or a hybrid capture panels, and followed by the sequencing through DNVSec platforms. And the data analysis can be done either by Megabolt or Gtron. Then once the variant calling file is generated, we partnered with third-party company to annotate and also interpretation of those variants and also report generation. Okay, so this way we are providing end-to-end solution starting from extraction to the report. So MJ is providing the one-stop package for all the applications, including precision cancer applications and also and also single cell and also spatial. So this way end-to-end workflows will be provided. Okay, either from MJ and also including the third-party company partnered with MJ. So coming to the publications, as I mentioned to you, we started manufacturing of the shorted sequences from 2017 onwards. So since I think nearly 8 years, we got more than 10,000 publications using the data generated by the DNVSec platforms. Most of these publications, they have been published in high impact journals. So here you can see in this slide, depending on the data output and also price per GB, the higher the data output generated by the instrument, the lower the price of the GB. Okay. So for example, as I mentioned to you, the T20, it is generating 72,000 GB data output in one run. And the price for 1 GB data output is only $1. So this way, okay. The higher the data output, the lower the price, not only for MJ, even from other companies also. So wherever the instruments are generating higher data, the price per GB will be low. So whatever till now we have discussed about what are the data outputs for different shorted sequences. Then what is the accuracy of the data? So for example, here you can see we are providing minimum guarantee of 85% of the bases to get quality score of Q40 or more. So generally the quality of the sequencing will be analyzed using a metric called FRET 33 score. So that will be generated in Q20, like Q10, Q20, Q30, Q40, like that. So Q40 means out of 10,000 bases, we can expect less than one base as an error. So that means the error rate is very low. Okay, very low. So that's the reason we are guaranteeing minimum 85% of the bases to get Q40 score or more. That means the very, very, very low error rate. That means the reports generated by the data generated by our DNBC platforms, the reports will be very accurate without any error rate. So here I'm just showing you some of the DNBC platforms which are very ultra fast platforms. For example, E25, G19 and T1 and also T7. These 4 platforms are the ultra fast platforms because the turnaround time for the sequencing within is less than 24 hours. Except T7, remaining all E25, G19 and T20, T1, they can complete the sequencing plus demultiplexing of the data also within 24 hours. Whereas T7, it takes additional 2 to 3 hours for the demultiplexing. That means 22 hours to 24 hours for the sequencing and additional 3 hours maximum for the demultiplexing. So in the T series, here I'm just showing you some of the highlights. Left side, you can see there are multiple companies for using running large scale omics research projects. For example, the Gene Plus, they are using the T7 platforms for their large scale omics project. And in Spain, you can see that there is one institute called the Karolinska Institute, which is using our G4 and also T7 since last I think more than 5 years. I think they have completed already near 0.5 million metagenomic samples using our G4 and T7 in Spain. So recently in the Indonesia, they have started a national genome project for the whole genome sequencing of the population. They are using our T7 platform for that. So the data generated by T7 and T20, so nearly 1,000 publications have been published. And right side, you can see some of the keywords. One of the keyword we are all know Aerophils genomics. So here you can see some of the testimonies not only from Asia Pacific region, even from France and also UK for the T7 platform, T7 and T20, but for the best performance. And because of the best performance of the T7 and T20, they have achieved multiple awards. Here you can see some of the awards. So regarding the T20, as I mentioned to you, it can run 6 flow cells at a time. But here, it will not give you the flexibility to run 1 flow cell or 2 flow cells separately because it is something called dipsec. So the sequencing reagent, all the flow cells, 6 flow cells will be dipped into the sequencing reagent one by one. So that's why the sequencing reagent, it cannot be reused after the one use. So that's why all the 6 flow cells must be sequenced in one run. So that's why we are able to provide less than $1 for 1 GB. So depending on the read length, it will generate either 48 GB TB data output or 72 TB data output. For example, if you run P150 flow cells, 6 flow cells, in 6 flow cells, it will generate 72,000 GB data output. That means you can complete the whole genome sequencing of 576 samples in one run. So this is really one of the best platform for those countries who would like to sequence whole genome sequencing of the population for huge number of samples, maybe 1 million, 2 million. Maybe we need to inform to our government of central government also, because they have recently completed 10,000 whole genome sequencing on, right? So maybe by using this T20, we can increase the number of samples for the whole genome sequencing, maybe 1 million or 2 million. And as I mentioned to you, the cost of the sequencing is very, very low. So we can complete within $100. That means within 8,000 rupees, we can complete the whole genome sequencing, targeting 100 GB per sample. So regarding the T7, which is already being used by Haystack and also 2 more companies in India, they are using this T7. So total 4 T7s have been installed in India. So the T7, it can run 1 flow cell to up to 4 flow cells. Each flow cell, depending on the data output, read length, it can generate 1.2 or 1.7. So if you run paid and 150 flow read length, it will generate 1.7 TB data output. So in 24 hours, by running of 4 flow cells, it will generate more than 7 TB data output, 7,000 GB data output. So that's why it will be highly useful for the service providers for having more than 1,000 samples every month, or so those clients who would like to run whole genome sequencing or deep exome sequencing targeting higher data, also epigenomics samples for the methylome sequencing, and also targeting the higher data of the whole transcriptome. For example, instead of 30 million reads, maybe those clients who are targeting 100 millions per sample for whole transcriptome, this T7 will be highly cost affordable for them. So apart from that, the T7, it is recently optimized for a company called Olink for the NGS-based proteomics. Please note, NGS-based proteomics, because this Olink company, it is having a panel called Olink Explore HT, which is covering around 5,400 proteins. So by using this panel, the library can be prepared and using the sample of plasma or serum, so we can explore the profile of the expression levels of these 5,400 proteins by using this T7 platform. So they have already optimized, and this T7 platform is being used for the NGS-based analysis, proteomics analysis of the 5,400 proteins. So this kind of number of proteins cannot be analyzed even by the MS. Okay, so we are providing the end-to-end solution, including the analysis and reports. So coming to the T1, this is the instrument of the T1, which is released just 1 month, 1 month back, I think, 1 month. So this is upgraded from the actually G400. Okay, so this T1 is also able to sequence either one flow cell or two flow cells. You can run one or two flow cells separately. Okay, so the T1, it is one of the ultra fast platform, because the sequencing runtime is less than 24 hours, even for the paid and 150 also. So in each flow cell, if you run E150 read length, it will generate 600 GB data output. So that means if you run two flow cells, it will generate 1.2 TB, 1200 GB data output in full run by running two flow cells. And what is the accuracy of the data? So we are guaranteeing minimum 90% of the bases will be having Q40 or more. So that means the accuracy rate is very, very high in the T1. So actually we are guaranteeing 90% only, but when we tested the performance, more than 94% of the bases are getting Q40 or more. Okay, I will show you in the coming slides. And apart from that, this T1 is having an advantage of this is having the integrated DNB making, because my colleague he will explain you why do we need to prepare the DNA nanoball and those things. So generally, once the library is prepared, once the library is constructed in the circulation, then we need to prepare the DNA nanoball out of the flow cell. Okay, so generally it takes manually. It will be done manually and takes at least half an hour. And so, but here this instrument is having the integrated DNB making. Once the libraries are prepared, once the libraries are converted into the circulation, then the pooled libraries can be loaded into the T1. Then T1 can take care of the DNA nanoball making and loading of the DNA nanoballs into the flow cell, and the sequencing will be started. We don't need to manually intervene into anything. So starting from the DNA making, loading of the DNA nanoballs into the flow cell, followed by the sequencing, everything can be done by the instrument itself. So that's why it will give you the high flexibility. And apart from that, we are providing two configurations. So those clients who would like to analyze the data also immediately, then we are providing the built-in bioinformatics server inside the instrument, that is called T1ARS. So this is having the built-in bioinformatics server inside the instrument. So that's why once the sequencing is completed, sequence control software itself, it will do the demultiplexing, FASTQ files will be ready. And if you have this built-in bioinformatics server, the secondary analysis also can be done by the instrument also very quickly. So this way it will be highly helpful for those clients who would like to generate the report very quickly. So here I'm just showing you how rapid this instrument is to complete the sequencing and also demultiplexing. So for example, here you can see the T1 maximum 24 hours for the PE150. If it is SC50, it is only 7 hours. There are other companies like Element AVT and also Illumina Nextec 2000. They take more than 38 hours to 48 hours. So this way, this instrument can save at least 20 hours of the time compared to the other companies. Okay, even the GeneMind nowadays, some companies they are taking the GeneMind Surface 5000. It also more than 7 hours higher. So this is the one, the integrated DNA making and also how it will be done. So okay, so once the libraries are converted into the circulation, then the pool libraries can be loaded into the instrument. And you can load either pooled single standard circulation library or double standard DNA library from Illumina or DNA nanoball making, either one. Okay, so if you load either single standard circular DNA, then the DNA nanoball making can be done by the instrument itself. Or if you load the Illumina double standard DNA libraries, then conversion into the single standard circular DNA followed by DNA nanoball, everything can be done by the instrument itself. So here I am just showing you starting from the extraction to the library analysis, because the built-in bioinformatics server is also there in the T1, right? So I'm just comparing with the other company platforms. For example, compared to the Nexec 2000. Okay, so in the T1, starting from extraction to the sequencing by the instrument and analysis by the instrument itself, it will be maximum 32 hours. Whereas the Nexec 2000, it takes up to 51 hours, because they are also providing onboard Dragon, but it takes, compared to the same data, it takes 51 hours. So that means here you can save nearly 20 hours of the time in the T1 if you purchase the T1. So what kind of flow cells are being provided for the T1? So we are providing very small flow cell generating 500 million reads, medium flow cell that is covering generating minimum around 1 billion reads, and flow cell large 2 billion reads, 2000 million reads. So depending on your number of samples or depending on your targeted data, you can decide either to use large flow cell or small flow cell or medium flow cell. And here you can see we are providing SC52 up to SC400, that will be released very quickly. So the very recent, so the P50 to P300. So this way we are providing in single end 50 base pairs to 400 base pairs, paid and 50 base pairs to 300 base pairs in paid end. So independent of either what kind of read length it is, we are guaranteeing minimum 90% of the bases to get minimum Q40 or more. Okay, whereas the P300, because of the large read length, generally we are guaranteeing minimum 75% of the bases to get Q40 or more. If you compare with other companies, no one is guaranteeing 75% of the bases to get the quality score of 40 or more. Most of them, they are guaranteeing only Q30 for the 75% of the bases, but we are guaranteeing Q40 for the 75% of the bases for PE300. Okay, so this is the way the quality of the score is, quality of the data is very high in DNBSEC platforms of MGI compared to the other companies. So when we tested the performance of the T1 with oncology samples using FFP samples and also liquid biopsy samples, so using a panel, okay, so that is the gene plus comprehensive genomic profile panel, 1021 gene. So this is the one. So comprehensive genomic profile panel, this has been used for the FFP sample and also liquid biopsy sample. So the data generated by the T1 is able to detect the variants which are having the variant allele frequency down to 0.5% in the FFP samples. Whereas in liquid biopsy, generally most of the variants will be having very ultra low frequency. So this instrument data is able to identify the ultra low variants which are having the frequency down to 0.2%. So this way we can identify ultra low frequency of the variants in the oncology samples, even in the liquid biopsy samples and also FFP samples, solid cancer samples also. So here you can see, though we are guaranteeing only 90% of the bases to get a quality score of Q40 or more, here minimum 94% of the bases are getting Q40 or more. And what about Q30? 97.78, almost 98% of the bases are getting Q30 or more. So apart from that, this T7 has been tested even for the single cell transcriptomics also. As I mentioned initially in the first slides, we have designed the droplet generators for the single cell transcriptomics, because once the cell suspension has been prepared, the cells, because here single cell means single cell RNA means each cell RNA should be captured from each cell and the data should be generated from each cell using the RNA molecules. Okay, so that's why each cell should be dropleted into the encapsulated into the droplet. So that's why we have designed two droplet generators. One is the C4 system, second one is the CTM4. So this C4 system is not required, it can prepare droplet generation only one sample at a time. Okay, whereas if you need to run multiple samples for the droplet generation, this automated droplet generation CTM4 will be helpful. It can run either one sample to up to 4 flow cells for the droplet generation. So once the cell suspension has been prepared, that cell suspension should be loaded into the microfluidic chip, because the microfluidic chip should be inserted into this one. I will show you that one. So this is the microfluidic chip which will be provided together with the kit. Okay, so the cell suspension should be loaded into this well, and also cell beads. The cell beads will be having the poly DT. So the poly DT will be used for the capturing of the RNA lysed from the cells, RNA released from the lysed cells, because generally RNA will be having the poly A tail, right? So that using that poly A tail RNA, this the cell beads which are having the poly A tail, they will be capturing the RNA which are having the poly A tail. So that will be loaded into these cell beads also will be provided into the kit. So then the oil which will be provided into the kit, that will be so then it will generate the droplets. Well, when the droplets are being generated, the cells and also the cell beads will be encapsulated within the droplet. Then those droplets which are generated, that will be collected into this one. So once the RNA is captured onto the cell beads, then the droplets will be breaked out, and then the reverse transcription will be done to convert RNA into the cDNA, and double stranded cDNA will be prepared. Then it will be fragmented. Then barcodes and adapters will be ligated to that one. Then sequencing can be started after the library protein is converted into the single stranded circular DNA and DNA nanoballs. Okay, so for the best performance of the results, we are suggesting the cell viability should be more than 80%, and you can load the input number of cells 5,000 to 30,000 cells into the chip. And please note that the cell diameter can be even up to 60 micrometer also. So our microfluidic chip, it cannot be clogged even with if you load the plant cells which are having a very high diameter, for example 60 micrometer, whereas other companies, I will not mention the name, so they are providing the chips which can which will be clogged if you load any cells which are having a diameter more than 30 micrometer. So that's why our microfluidic chips will be highly useful to capture the RNA. When we tested the performance of the T1 for the single cell using the PBMC cells, here you can see for each cell we have generated 23,000 of the reads. Okay, so depending independent of this 23,000 reads only per one cell, the identified number of median number of genes per cell is more than 4,000, and apart from that, more than 15,000 of the transcripts have been identified in one cell. So this way, if you see the results compared with other companies, with 23,000 of the reads per one cell, the results are really very, very high compared to other company results with the same number of PBMC cells and with 20,000 of the reads per cell. And if you compare with what is the count of the transcripts per cell and count of the genes per one cell is very low in other companies. But here, minimum 4,000 genes can be identified in one cell. And also number of transcripts per one cell is 15,000. So apart from that, this instrument has been tested even for the spatial transcriptome also. So our technology that is called stereosec. So here, once the tissue section has been prepared, the tissue section should be mounted onto the chip, because this chip will be having the DNA nanoballs will be deposited under the silicon chip. So this DNB nanoball will be having a coordinate index, okay, spatial coordinate index. So this coordinate index will be used for the getting the information, spatial information of the transcript. So where this transcript is coming from the tissue section. So this way, by using this coordinate index, the spatial information, localization information of the transcript or cell will be collected from the tissue section. So billions of the DNA nanoballs will be deposited on the chip. So then each DNA nanoball, I will show, each DNA nanoball will be having coordinate index for the collecting of the localization information of the transcript or cell, and also the UMI, unique molecular identifier. This will be used for the identification of unique transcript coming from every cell, and this poly-DT will be used for the capturing of the poly A coming from every cell. Okay, so whereas if you need to run FFP samples generally RNA will be highly degraded. So then the poly-DT will not be helpful for capturing of the RNA coming from degraded RNA. So that's why for the FFP samples we are providing the random primers instead of the poly-T to capture the RNA from degraded RNA from FFP samples. Okay, so this is the chip. So this is one of the chip. The field of view is 30 square centimeter. So that means you can mount the large tissue sections like human heart or human brain also. So once the tissue section has been prepared and it can be mounted onto the chip, then you need to stain it with either H&E or DAPI or single stranded DNA staining. We are providing ssDNA staining because this ssDNA staining will be helpful to automatically get the cell segmentation. So once the cell segmentation has been prepared, then you can identify what are the DNA nanoballs covered for one cell. So then once the imaging is taken, then using the permeabilization, the RNA will be captured onto the DNA nanoball using this poly-DT. Then that will be converted into the single stranded cDNA using the reverse transcription. Then the double stranded cDNA will be collected. Then after the fragmentation, adapter plus barcodes will be ligated. Then they will be converted into the adenine nanoball. Then sequencing can be done either on T1 or T7 or G400. Okay, so these are the chips which will be provided. So here I am not there is one slide which is missing here, that is 0.5 square centimeter chip, which will be used for the small tissue sections. So starting from 0.5 square centimeter to up to 30 square centimeter we are providing. So this kind of huge chips are not being provided by any other companies. So when we tested the performance of the T1 for the spatial transcriptomics, here you can see there is something called bin20 cell binning. So bin20 equal to the 10 micrometer of the cell. So in generally human samples will be having a cell diameter of 10 micrometer to 20, right? So in one cell, we are able to identify, by T7 data is able to identify more than 477 genes per one cell. Okay, and the transcript number of transcripts also nearly 600 plus. Okay, so if you see the results, we are able to provide the high resolution down to single cell, and even single cell also we are able to identify more than 400 genes per one cell. So this kind of results are not being provided by even other companies which are using the same kind of technology, like chip based analysis for the spatial transcriptomics. Okay, so that's why we are able to provide high resolution down to single cell. Each single cell you can identify 400 plus genes per one cell, and also the higher number of transcripts, and together with the spatial information of the each transcript coming from tissue. So G400, I think most of you are already aware of this G400, because here you can see because the G400 is being used by many universities, service providers, and also hospitals across the world. So that's why there are multiple testimonials because of very high performance of the G400 since last 8 years. So here also it is we are providing small flow cell to large flow cell. So I'm not going to the details because of time limit. And here I would like to show one more platform which is released just 8 months back. So because this instrument is designed for the histopathology. Okay, generally the conventional histopathology tool is immunohistochemistry, right? So for the tumor microelement analysis, because once the tissue section has been mounted on the slide, then imaging, after the immunohistochemistry staining, the imaging will be taken. Then the pathologist, they will analyze the cell morphology and tumor classification, and where the cells are organized into the tissue section. That means the spatial organization of the cells for the cancer diagnosis and progress treatment. So this way it is very labor work for the immunohistochemistry. But nowadays there are emerging developments in the histopathology, including the automation, multiplex, and spatial information. So I think because of the time limit, I need to quickly complete. So here I'm just showing the comparison of conventional histochemistry and multiplex. So in the conventional immunohistochemistry, you can identify only one protein marker in one tissue section, whereas if you need to identify multiple protein markers, it's really not possible. You need to serially prepare multiple tissue sections, and each tissue section can be used only for one protein marker. Whereas nowadays multiplex immunofluorescence that is being used for identification of the up to 24 markers in one tissue section only. You don't need to run multiple tissue sections. So that's why we have designed the G400 system only into a Fluo Expert. So this instrument is nothing but it's G400 only. So the instrument is converted into the automated staining and automated image taking of the multiplex immunofluorescence. So once the tissue section has been mounted onto we are providing these flow cells, okay, these chips. So once the tissue section is mounted on the chip, then the chip can be loaded into the instrument, and the automated staining, automated image taking, and automated analysis. Everything can be done by the instrument itself. I will show you the. So you can use this instrument either for staining for the. So that's why this will be highly useful for the diagnostic companies for getting histopathology samples for the digital pathology. So whenever you need to run the multiplex immunohistochemistry, immunofluorescence, then you can switch the software from sequencing to staining. Or if you need to use the instrument for the sequencing, you can switch the software from staining to sequencing. So this way, this instrument will be used for either sequencing or staining by switching the software. So here you can see the workflow. Once the tissue section has been mounted on the flow cell, then the dewaxing, rehydration, and then it can be loaded onto the instrument, Fluo Expert. Then staining and everything will be started. Okay, so everything will be done automatically. So how long time does it take for the one protein marker? For example, it can run up to 24 markers. Okay, so for one protein marker, it takes 50 minutes only. Whereas the conventional immunohistochemistry takes 3 hours for one. So you can save a lot of the time. And if you need to identify 24 protein markers, it takes maximum of 24 hours. That's all. So the instrument software, the Fluo Expert Visual Analysis software, it can do the automated analysis also, visualization and quantification of the expression levels of the protein markers from one protein to up to 24 proteins, and cell segmentation. And if you need to identify the spatial analysis or spatial organization of the cells and cell types, that also can be done by the instrument software. So we are providing 3 chips: small, solo, that means the tissue section, if it is small, you can load it on the solo small, and if the tissue section is large, you can load it on solo large. And if you need to run large tissue sections, you can use this dual large. Okay, so you can use either fresh frozen tissue or FFP blocks also for the mounting of tissue section onto the chips. So we tested the results generated by the multiplex immunofluorescence by Fluo Expert compared with the gold standard immunohistochemistry. So for example, here you can see the CD3 protein expression level has been quantified by Fluo Expert 34.64%. So when we tested with the gold standard with the same tissue section, it is 34.27. That means results are almost 100% same with the gold standard immunohistochemistry. That's why the results are very highly accurate compared with the gold standard immunohistochemistry. Okay, so how many samples you can run in 1 week? So if you use the dual large, you can run up to 140 tissue sections in 1 week. So these are all about DNA nanoball platform using the DNB-seq technology. So coming to the cyclosec long-read sequencers, I will skip some of the slides because of the time limit. So by using this, our cyclosec platforms, they can be used for any kind of clinical research projects or scientific research projects also. For example, the tumor detection and HLA typing, and even rapid pathogen identification. I will show you in the coming slides. And for the scientific research, for example, de novo genome assembly of the human genome, any kind of whole genome sequencing, or methylation analysis and transcriptome analysis and chromatin structure analysis, even the single cell and spatial transcriptomics also. So all applications can be supported by the cyclosec long-read sequencer. So we have designed first platform is WT02. So it can run either 1 or 2 flow cells. Okay. So I think this principle, my colleague, I think Harald, will explain. So I will just briefly I will explain this one. So each chip of the cyclosec WT02, it will be having 1024. So these nanoproteins will be deposited on the membrane. So this chip will be submerged into the electrophoretic system. So then a current voltage will be applied on entire chip. Okay. So once the DNA is extracted, the DNA should be converted into the library. So we just need to add a adapter sequence to the DNA, because the adapter sequence, this motor protein, that will be binding to the adapter sequence. So then what it will do is, the motor protein, it will unwind the double stranded DNA to the single stranded, because this nanoprotein will allow only the single stranded DNA molecule to run it through this hole. Okay. So that's why this motor protein, it will be binding to the adapter sequence of the DNA, double stranded DNA. Then it will unwind the double stranded DNA to the single stranded. Then it will be moving the single stranded DNA molecule one base by one base through this nanopore hole. So as I mentioned to you, a current voltage is applied, right? So when the base is being translocated through this hole, the current fluctuation will be there. The current fluctuation will be very specific to every base. ATGC will be having different current fluctuation signals. So each channel will be connected to the electrode and sensor. So when the base is being translocated through this channel, the current fluctuation will be generated. That will be identified by the sensor. That sensor will transfer the information of the current fluctuation signals to the instrument software. Then software, it is having a base call algorithm that is on the basis of the automated speech recognition. Then it will identify on the basis of the current fluctuation signal, it will identify what base it is, whether it is A or T or G or C. Yes. Yeah, I've completed almost. Yeah, it's specific to every base. That's why the base color algorithm, it will identify the current fluctuation signal, and it will identify the base. Okay, so I will skip these things, I think, because of the time limit. So so we are providing the Bioinformatics server also, which is providing the mapping aligners and also de novo assemblers also, and variant colors. So when we tested the results, so here you can see the results are coming from the read length is 100 base pairs to up to 1.1 MB, and the speed of the sequencing by the WT0T is up to 420 base pairs for 1 second. So if you run it continuously for 72 hours, it will generate 50 GB data output. Yeah, completely. Yeah, I will skip these of some slides. Okay, so I'm just showing you some of the applications which are already tested on the Cyclosec. Here you can see there is something called the human de novo genome assembly. So there is something called Contig N50. So if you see the Contig N50 for the whole genome sequencing of the human sample, it is 33 MB. It is almost equal to the PacBio. And later, so for the metagenomics, when we tested this performance, so this data generated by Cyclosec is able to identify all the species, and apart from that, the data has been successfully used for the construction of the genome assembly of every species. If you see the coverage of genome assembly constructed by the data generated by Cyclosec, it's almost 100% for every species. So that's why this data generated by Cyclosec is highly helpful for you. So, and rapid identification of the pathogens. So here we tested the performance of the instrument with 1 minute data versus 2 hours data. So when after mapping of the 1 minute data and also 2 hours data with the reference genome, all the species of the fungal species have been successfully identified even with 1 minute data. So that's why we can very rapidly identify the species by using the Cyclosec platforms. So the telomere to telomere genome assembly also can be constructed by the plant samples for the instrument. So here you can see for the animal, the Contig N50 is almost 274 MB. Yeah, so here just two slides are there. So we are trying to authorize one of the service lab in India for the DCS. That means for those clients who are not able to purchase an instrument, they can outsource the samples for the DNA or RNA sequencing or single cell omics sequencing or spatial omics sequencing.
Thank you so much. Thank you, everyone. I conclude the session now.